Let's now switch to regulatory networks. So the next kind of network we'll talk about are regulatory interaction networks. And so these networks are pretty easy to understand because we already did some lectures on like ChIP-seq and sequence analysis. But basically, again, the idea of regulatory interaction is that the nodes in your network represent either transcription factors or regulators in general, as well as target genes. And in these regulatory interaction networks, you're basically drawing edges between regulators and the genes or the targets that they regulate. So oftentimes, if you have an edge between a transcription factor and a target gene B, that usually represents transcription factor A bound to either an enhancer or promoter that regulates the expression of gene B, and that's what those interactions represent. And so by and large, regulatory interaction networks are usually generated through using ChIP-seq. And so we already went through what ChIP-seq does, but essentially ChIP-seq allows you to sequence reads that correspond to binding sites or near close to binding sites of a particular transcription factor. And then hopefully if that regulatory element that's bound by the transcription factor is close to the target gene, then it's easy to identify what gene that transcription factor targets. Some of the pitfalls of using ChIP-seq to characterize regulatory interaction networks are, of course, number one of the biggest problems is that if you actually look at any real ChIP-seq data, for most transcription factors, a lot of their binding happens outside of the promoter region. And so then that really begs the question, well, sure, you can use ChIP-seq to find the binding site of a transcription factor, but that doesn't actually necessarily tell you what gene it regulates. And so oftentimes people make an assumption like, well, if my ChIP-seq experiment tells me that TF binds to a, say, non-coding region of the genome, I'll just look to see what the nearest gene is, where the nearest transcription start site is, and I'll just assume that that's a target gene. That turns out to be wrong oftentimes because of looping and things like this. Some of the other pitfalls of ChIP-seq we already talked about. So you need antibodies, generally speaking, and those can be really hard to make, especially good antibodies. And because transcription factor binding can be cell context specific, then even for a single TF, to really understand where it binds and under what conditions it binds, you'd have to perform ChIP-seq assay for like the thousands of types of cells there are in, for example, the human body, just for a single TF. And so that's not really scalable. And so I showed a figure like this in one of the earlier lectures, but basically here's a figure on the bottom showing you a map of different cellular contexts as rows and different transcription factors that are being ChIP-seq as columns, as well as some histone modifications. And basically a green dot indicates that transcription factor has been ChIP-seqed in a particular cellular context. And the main point here is that this matrix you see on the bottom of the figure is more or less mostly white. And it's mostly white because even for this ENCODE consortium that is possibly the largest consortium of people doing ChIP-seq experiments in human and mouse, even for them, they are not able to ChIP-seq most TFs in most cellular contexts. And so that should just give you an idea of how sparse or how hard it is to generate these regulatory networks. So one of the less popular ways of generating regulatory networks is through position weight matrix scanning. And so we already kind of went through how to construct position weight matrices and how to do a basic scan using them. Here I'll just talk a little bit about pitfalls of using them to generate regulatory interaction networks. And so the first major problem you have is that there's a lot of degeneracy in transcription factor binding site recognition sequences. And so, like I mentioned before with PWM scanning, PWM scanning is good at detecting in vitro binding sites, but not so much in vivo binding sites because there's a lot of in vivo, there's a lot of other factors that influence whether TF binds or not, like whether or not the chromatin is open at that particular position or whether or not there's other cofactors that are present that facilitate binding. One of the other big problems is that PWMs are not actually unique to any given transcription factor. So lots of transcription factors form families of transcription factors because they have related protein sequence and structure and function. And so here, for example, I'm showing you 3 different transcription factor families and different members of that family, as well as their kind of known binding sites or known position sequence logos. And you can kind of see that different members of the same family can have very similar sequence logos and position weight matrices. And what that means in practice is that it's actually really hard to distinguish the binding sites of these different factors. And in fact, in reality, within a given family, you might have a lot of cross-binding of one transcription factor binding to the binding site of another member of the same family, but just at like different binding affinity. And so, it's really hard to predict differences in binding of factors of the same family. And even factors of different families that share, say, parts of DNA binding domains and so on, a lot of their binding sites look pretty similar. Number 2, there's a big question that we didn't really talk about, which is that it can be challenging to figure out which position weight matrix to use. And so in the Chipseek lecture, I basically mentioned briefly that there are many databases out there where people have taken Chipseek data or selects data or PBM data and assay different transcription factors to generate sequence logos. One issue is that there's a lot of kind of technical variability in between the different assays that you can use to generate position weight matrices. Even for Chipseek, like depending on which cellular context you Chipseek a particular TF in, you might get a different set of binding sites, and that might lead to different position weight matrices. And so here, for example, under number 2, I'm showing you 4 different PWM or 4 different sequence logos and respective PWMs for the same transcription factor, UBX, that's found from different websites. And you can basically see that even though they all kind of qualitatively look similar, if you were to actually use these 4 different PWMs or sequence logos to actually scan and search for UBX binding sites, they would actually return different hits, just because the specificity at each position is actually quite different between these 4 different PWMs for the same factor. Again, number 3 basically refers to the fact that binding in vivo is often context dependent, and PWMs currently do a poor job of capturing that context dependency. Number 4, PWM libraries oftentimes are really incomplete, and so in the human genome, for example, depending on how you count it, people think there's roughly like 3,000 DNA binding transcription factors, most of which we don't have a PWM for in the first place because someone just wasn't able to, mostly because someone either wasn't able to generate a good antibody for each one of those TFs, or or those TFs just haven't been studied yet.